Hello everyone. Am I audible to all?
Yes, ma'am.
Yes,
Yes, ma'am.
Yes, ma'am.
yes, ma'am.
Yes, ma'am.
Thank you so much. Welcome to our captivating webinar on human sperm vitrification, exploring recent breakthroughs, presented by Heredity Biosciences. I am Srishti Swagatika, your coordinator for this enriching session. Join us to unravel the wonders of cutting-edge fertility advancement. Delight is an educational journey as we uncover the secret of preserving life with simplicity and expertise. Heredity Bioscience, a renowned biotech company and research center in Odisha and Guwahati, proudly brings you this enlightening experience. Before we proceed, I'd like to introduce our esteemed speaker for today, introducing Dr. Feng Gao, a distinguished senior clinical embryologist with a profound passion for advancing assisted reproductive technologies. With a wealth of expertise and research contribution in the field of Dr. Feng Gao remains at the forefront of revolutionary fertility treatments and bringing hope to countless individuals and families worldwide. We are fortunate to have her share her insights with us. I would like to take a moment to introduce our esteemed convener of the day, Dr. Gopal Krishna Purohit. He is the CEO and co-founder of Heredity Bioscience, and he brings with him a wealth of knowledge and experience in the field of clinical genomics and infertility research. We are honored to have him leading us within this webinar. Before we proceed further, I would like to share some important information about the schedule and the instruction for today's session. We kindly request all the participants to stay until the end of the webinar. At the conclusion, we will provide a feedback link to you share your thoughts and suggestions. Please note that fully completed feedback forms will be considered for certificate. Your feedback is valuable to us and we appreciate your cooperation in this regard. Also, it comes to asking questions or participating in discussions, we kindly request that you maintain discipline and respect for others' opinions. This will ensure a smooth and productive exchange of ideas. Now, without further delay, I am delighted to hand over the virtual stage to our director, Dr. Gopal Krishna Purohit. Once again, a warm welcome to all the participants. Let's make the most of this educational opportunity and learn together. Thank you, and over to you, Dr. Purohit. Please, sir.
Is it my slide visible?
Yes, sir.
Yes, sir. Yes, sir.
Great. Thank you. Good morning, everyone. I'm thrilled to here today as we begin on this exciting journey into this reproductive technology. I want to express my heartfelt gratitude to all of you for joining us in this webinar. Before we dive into this heart of our discussions, I would like to extend a special thanks to Susri for a kind introduction. It's an honor to be a part of an esteemed event and have the opportunity to share valuable insight with all of you. I am Dr. Gopal Krishna Purohit, CEO and co-founder of Heredity Biosciences. It is a pleasure to have each and every one of you here with us. Today's webinar focus on this topic, human sperms vitrifications, of recent process. This subject would emerge important as we explore the cutting-edge advancement in reproductive technology and potential its hold for advancing infertility challenges. The techniques of sperm vitrifications has revolutionized assisted reproductive technology, offering hope and viable solutions to couples seeking to start a family. I want to express my deeper gratitude to our guest speaker, Dr. Feng Gao, from San Francisco IBI RMA Global, who serve as a senior clinical embryologist for accepting our invitations to be today's guest speaker. Dr. Feng Gao's expertise and knowledge in the field of fertility are truly commendable, and we are honored to have here share her insight with us. Her valuable contributions to today's webinar will undoubtedly enrich our understanding of human sperm vitrification and recent advancement in reproductive medicines. Thank you, Dr. Feng Gao, for being a part of this informative event. Ladies and gentlemen, fasten your seatbelt as we embark on this exhilarating journey into this world of human sperm vitrification, review of recent progress. In these presentations, our esteemed guest of speaker, Dr. Feng Gao, will lead us through the cutting-edge field of infertility issues, sperm preservation techniques, and the latest success rate in assisted reproductive technologies. We will uncover the on-background advancement that revolutionized the way of approach fertility challenges and discover the immense potential these techniques hold for individuals and couples worldwide. Dr. Gao's expertise and knowledge in this field are truly commendable, and we are honored to be here with the invaluable slides with us. So once again, thank you, Dr. Feng Gao, for gracing with us. Your presence today, we greatly appreciated your times and knowledge. As we begin this webinar, let us emphasize the utmost importance of this subject at hand. During this session, our esteemed guest speaker will shed light into the revolutionary techniques for human sperm vitrification as an innovative method for sperm freezing that achieves cryopreservation through the use of ultra rapid cooling rates, preventing ice crystal formations. Before the process to share the slides with our guest speaker, I would we addressed the significant highlights in the history of advancement in enriching the in vitro fertilizations. This include commentating the birth of the first tasty baby, a pivotal milestone in the field, and discussing the latest progress made in the ever-evolving domain of reproductive technology. Now, a subject that is the deepest of the human heart, that is infertility. Infertility is a condition that affect many couples worldwide, causing significant emotional and physiological distress. It refers to the inability to conceive a child naturally after a year of regular unprotected intercourse. It is a reminder of the complexity of this life and the profound desire for parenthood and adults. Understanding the challenges couples face this infertility is crucial to appreciate a significant of Louise John's path through IVF. Everyone though knows the picture of this beautiful lady, might be Louise Johnson. She is a world's first IVF baby. Her birth on July 25, 1978 stand as a momentous achievements in the field of reproductive biology, forever challenges in the landscape of infertility treatment. Louis' entry into this world was a testament to human perseverance and scientific innovations conceived through in vitro fertilizations. Her birth was the conclusions of the pioneering effort of British gynecologist Patrick Tuppy and scientist Robert Edwards. Before Louis' history arrival, couples struggling with infertility faced limited options and heartache. IVF opened a new chapter offering hope and a potential solutions to countless families, from the child to their own. The groundbreaking achievements paved to the way to the further advancement in reproductive technology. Today, IVF has become a worldwide embraced fertility treatment responsible for millions of births worldwide. Louis Joyce Bond's life was been an inspiration of hope, inspiring countless individuals to believe the possibilities that science and conversations can create. Her story continues to touch heart and speak conversations about the potential of assisted reproductive technology. Over the year, the field of in vitro fertilizations has witnessed remarkable progress and transformative achievements. These breakthroughs involvements, including multiple follicles in the mother's ovary using human monopocal gonadotrophins, leading to the retrieval of pre-ovulatory oocytes. The oocytes were then fertilized in vitro and resulting embryos were carefully transferred to the mother's uterus. on their day 3 and day 5 transfer. Since then, the practice of IVF has continued to evolve at the astronomical pace. Today's IVF astronoming the millions of births worldwide and play a vital role of all countries. The growing demand of fertility treatment drive constant research and development to optimize IVF protocols and success rates. In the majority of IVF cases, infertility couples undergo treatment to conceive genetically related children, bring joy and fulfillment to families. Moreover, IVF has become a valuable tool in genetic testing, allowing couples to reduce the transferring of single cell gene mutations associated with the health concerns. As you reflect to these milestones, as we reminder of the immense impact of IVF as an individual and couples seeking to the build of their families. The utilizations of donor sperms and oocytes has become increasingly common, widening the possibility of hopeful parents. Additionally, advancement have allowed women who have unable to carry to pregnancy to turn to gestational career as a viable options. Now, audience, before we begin to our webinar and introduce our esteemed guest, allow me to provide a brief overview of Heredity Biosciences. Today, I am thrilled to present the journey of Heredity Biosciences, a leading provider of research and training services to hospital and life science institutes and pharmaceutical companies, established in 2018 and reconstructed in 2021. Our company has gathered trust and recognition among doctors and research scientists. Backed by a team of experienced professionals in life sciences, healthcare, analytical and metabolic services, we are committed to delivering a top-notch solutions and innovation products. I'm excited to announce the inauguration of our new branch, Guwahati, which marks a significant milestone of Heredity Biosciences. At Heredity Biosciences, our unwavering commitment to scientific innovations drive forwards. Our team also committed to the research and innovations, continuing to explore some novel experimental methods to offer values insight into various life sciences and healthcare related aspects. Our diverse portfolio for services encompasses genomics, proteomics, metabolomics services in life science domain. We offer pan-India services, ensuring accessibility to our client access to the country. We focus on the crucial areas such as clinical microbiology, infection genetics, cancer genetics, fertility genetics, conventional PCR-based DNA studies. Through these services, we aim to contribute to the understanding, prevention, and treatment of various diseases and conditions. At Heredity Bio Academy, we are dedicated to providing top-notch training and research opportunity in the field of life sciences. At Odisha, we are leading training institute. We take immense pride in offering comprehensive hands-on experimental research training in both student and professionals. Our training modules over a wide range of interest for the field of including clinical microbiology, genetics, clinical microbial nanotechnology, genetics, food microbiology, genetics, and many more. These modules are designed to equip to participate to the necessary knowledge and practical skills required for this chosen area of expertise. In addition, our existing modules, we are thrilled to announce to introduce new specialized modules such as clinical microbiology and genetics, histochemistry and immunohistochemistry. These cutting-edge modules address to the evolving need for and advancement in the critical field, ensuring our participants stay in the forefront of scientific methods. Now let's talk about the expansional infrastructure facility at Heredity Biosciences that support our training and research endeavors. Both Bhubaneswar and Guwahati sites, state-of-the-art facility designed to provide a comfortable and conducive working environment for researchers and trainees. Our laboratory is equipped with world-class centralized air conditions, advanced equipment, and technology and facility research and experiments. Our facilities house comprehensive resources, including literature and research databases, empowering our participants with a rich learning experience. Our team of well-trained and experienced trainer and demonstrator offer invaluable guidance and mentorship, enhancing the learning journey. The Genomic and Microbiology Laboratory will equip with a support various research innovatives. We prioritize practical hands-on learning, providing ample opportunity for a participant to engage in hands-on experiments, fostering their practical skills and independence. Furthermore, we extend the publication assistance for those who wish to their research finding with the scientific community, promoting the culture of knowledge, disseminating and collaborations. We invite all these interested individuals to visit our central facility and witness the exponential infrastructures that support the research and training activities. We are confident that you will be impressed by the resources and opportunities available to enhance our scientific pursuit. So at Heredity Biosciences, we are thrilled to have the remarkable milestone achieved by the past 5 months. We are immersed pride to the providing valuable opportunities of research to students and professionals in the field of biosciences and clinical sectors. This is the highlight of our recent accomplishments conducted for this more than 500 students, both Bhubaneswar and Guwahati sites, one-day training program, more than 300 students successfully completed short-term and long-term intensive program, gaining hands-on experience. Our students have excelled in the placement activities with many securing positions in reputed organizations, setting the stage of this promising career. Most of the students joining various fertility clinics. With over 50 published research paper, our dedicated team has shown unwavering commitment to advancing scientific knowledge in biosciences and clinical sector. Strong collaboration with the esteemed hospitals like SCB, KIMS, and some hospitals have facilitated meaningful connections between academy and healthcare, leading to the experimental research to address critical healthcare challenges and enhance the patient care. Now the time for introduce our guest of speaker. It is with a great pleasure that I introduce our distinguished guest speaker for today's webinar, Dr. Feng Gao. Dr. Feng Gao holds a PhD and is a certified molecular biologist by the American Society of Clinical Pathology and the technical supervisor by the American Board of Bioanalysis. Dr. Gao's education from the prestigious University of California, Davis. Her academic journey laid the foundations of a remarkable career in the field of reproductive medicine. With a wealth of experience, Dr. Gao has played a vital role in various renowned institutions. She served as a molecular biologist at Chori, a visitor scientist at Lori Brackney National Laboratory, and contributed to the development of the Bristol-myositis peripheral blood cell capture systems at Bristol Limited, which is used for non-invasive parental testing. Her dedications to research and expertise lead her to serve as an assistant staff scientist at Children's Hospitals, Oakland Research Institute, and the clinical embryologist at CARE for the Bay Area, where she performs all aspects of chemistry, andrology, and embryology under the supervision of the lab director. Dr. Gao professional journey contributed a flourish as the assumed the role of Chief International Officer at HQA Fertility Center and as an embryologist at CCRM San Francisco and Nova Ardis. Currently, she holds the esteemed positions of Senior Embryologist and Research Scientist at IBI RMA Global, where she continues to contribute to the advancement in reproductive medicine. Her advancement have been recognized through several honor and awards, include the first prize at the annual meeting of Shanghai Pharmaceutical Research and Natural Science Foundations at Shanghai Block Grant and AHA pre-doctoral fellowships, among others. Dr. Gao possesses a diverse skill set, including expertise in mammalian cell culture and assay, ELISA, FACT, PCR, sequencing, molecular biology, and cell biology. Her hands-on experience with advanced technologies like the drop single cell manipulator systems and confocal microscopy shows her commitment to cutting-edge research. Furthermore, Dr. Gao has made significant contributions to the scientific community through her publications to top ranging from blastocyst formations to cholesterol regulations and human sperm vitrifications. With her extensive knowledge and expertise, Dr. Feng Gao bring invaluable insight into our webinar today. Please join me. Extended welcome to her as a remark on his journey and learning and diversity.
All right, so should we start over? Okay. All right, hello everyone. Sorry about that. All right, can we do next slide, please?
Okay.
The format is a little bit, but that's fine. A little bit off, but that's fine. Next slide, please. Yes, sperm vitrification usually when we load to the straw or cryo vial or something really small loading volume is usually about 1 microliter or 5 microliter because we need to achieve extremely high cooling rate from liquid phase to glass phase really quick. So the purpose of the human sperm vitrification usually used to preserve the fertility of the males who have a severe oligospermia or azoospermia patients. Next one, please. Next slide. All right, so yeah, I talked about the size. This is also giving you guys a little bit background about the size of a human sperm versus egg. The egg is about the diameter of the human egg is about 100 micron, and then also has a lot of cytoplasma compared to the sperm. When you look at the sperm, it's a very small, not much cytoplasma, not much water inside. Also, sperm has a large cell surface area. That's why when we freeze sperm or eggs, we have to use a different method because sperms are very sensitive to a high concentration of cryopreservation. When you look at the figure B, you can see that's under microscope, it's 8400 magnification. You can see the size about the sperm head is that's a 2 micron. The whole head will be like a 5 micron. When we freeze egg or embryo, we usually freeze individually, like one embryo, one straw. But for sperm, we usually freeze. For the slow freezing, we usually use a volume like a 0.5 mil or something. For vitrification, usually we freeze like a 5 or 10 together in a small drop. Next slide, please. All right, so principle of human sperm cryopreservation based on the thermodynamics. I'll explain to you guys a little bit later. Just I mentioned to you guys before, the sperm are very sensitive to exposure of a high concentration of cryoprotecting agents. That's why when we freeze human sperm, it's significantly different from those solution for oocyte embryo. And next, please. Next slide. All right, so here is just a process how we freeze embryo. Usually when we freeze embryo or eggs, we use two different solutions. First one we call the equilibration solution. It contains glycerol, ethanol glycol to use those permeable cryo solution to replace the intracellular water. Usually we call that ES media. We put there in 10 minutes, 10 to 12 minutes. Then we transfer second solution we call the VS. That's for vitrification solution. Usually we add sucrose in the solution. Sucrose is not permeable, cannot penetrate through the cell's surface membrane. So when they add sucrose in the solutions, usually it's a hypertonic suit. They can draw the more water from the cell inside. So once we move from ES to VS, ES usually stay in 1 minute, and then we load it to straw. Then we plunge into the minus 190-degree liquid nitrogen, and then we put the cap back and then put in the liquid nitrogen tank. That's usually how we do embryo cryopreservation. And next one, please. Next slide. This is a thermodynamics illustration. When you look at the picture of the top is liquid phase, green, as the temperature is above 0. You can see that's a liquid state. And also the transition from the liquid to glass phase, the temperature usually is minus 130 or minus 120 degree. So during when you drop the temperature from 0, especially like a minus 15 to minus 60, that's just called the super cooled phase. During this phase, ice crystals are really easy to form and can cause the damage to the cells. Vitrification, the principle of the vitrification is based on, so we try to achieve, once we have a liquid, we go straight to the glass phase, try to avoid or minimize the risk for the ice crystal formation in the super cooled phase. We just skip that because we achieved a really high, extremely high cooling rate. Next slide, please. So when during the cryopreservation, the ice crystal is the enemy for the cells. One thing is you can see this crystal directly can cut cell membrane and cause the cell injury. Also, intracellular crystal can also hurt the delicate structure inside cells and cause the death. The other thing is when you have a super cooled state in that, especially minus 15 to minus 60 degree, it's a super cooled phase. The outside of the cells is solid, and then it's a hypertonic. There's no liquid in there, so they're gonna draw more cells from the inside. Then they cause the dehydration of the cells and leading to the death of the cells. So that's ice crystal is the top of enemy for the cryopreservation. So vitrification is minimize the ice crystal formation. Next one, please. All right, so this one's I mentioned to you guys, so when we talk about sperm cryopreservation, usually we talk about two different methods. One is we call slow freezing, the other is vitrification. Slow freezing is commonly used every day. When we do sperm cryo, we're using slow freezing. Commonly, cryopregnants, usually we call it CPA. The CPA usually is a glycerol as 5 to 10%, and then sometimes the company has a test the yolk buffer. They add a glycerol and also the yolk. It's kind of a sperm membrane extender to help the survival rate, also help the motility recovery from the thawing. The procedure of the slow freezing is, so when you have the CPA media, you add it to the liquefied semen, a 1-to-1 volume ratio. You do very slowly, stepwise. And then once you mix thoroughly, you put the refrigerator like 2 to 4 degree for half an hour or 1 hour. And then you place to a vapor phase in the liquid nitrogen tank. And then after half an hour, and then you put in the liquid nitrogen. So it has been standardized for this protocol. And the device for slow freezing is we call cryovial. And also it has been approved by FDA in the United States. It has been validated for human sperm cryopreservation and storage. Usually the purpose of the slow freezing is for patient population is when they have a normal sperm volume concentration and sperm number, we usually do slow freezing. So for our clinic average, we do slow like sperm cryo every day. We have 2 or 3 cases we do. For vitrification, for our clinic, usually we have I think we started doing sperm vitrification about 3 years ago. The first year we do total like 5 cases, and then second year probably 10, and this year about same because only half a year. But we do see the trends of the human sperm vitrification going up. Usually, for the CPA for vitrification is a glycerol. Some vitrification methods are free of a permeable practicants too. The procedure for vitrification is, this is just a summary. I think it really depends on different device using for vitrification. In general, usually you put in the 37 degree for 5 minutes, and then you load a very small volume. Either put the cryo valve or the cryo loop or different device, and then plunge into liquid nitrogen. And it's not the standardized protocol. Really depends on different company, different device. So in United States, there are different device. Sperm VD is is the most popular one. I'll give a more detail about this device. This vitrification hasn't been approved by FDA. Also hasn't been validated for sperm cryopreservation and storage. So a lot of research and validation study needs to be done to validate it, because we don't know after vitrification and warming, what about the mitochondrial activity? What about the epigenetics? Is something change? I think that's our future study direction. Also, as I mentioned before, vitrification usually for the patients with a very small number of spermatozoa. All right, next slides, please. This is a cryopractants we call CPA in short. There are 2 different CPA. One is a permeable, is including glycerol, ethylene glycol, or 1,2-prop-deal, and the DMSO are typically used for cryopreservation. Non-permeable, usually including sucrose, glucose, or trehalose. Those are large molecule, cannot penetrate the cells. The reason why we need the non-permeable CPA, because it's stay outside of the cells, and hypertonic situation can draw more cells, more water from inside the cells. It's a kind of dehydration a little bit more, and make sure the permeable CPA can be equilibrated inside the sperm cells. All right, next slide. This is one of a study is developed by Dr. Zhou in China. What he was doing is, so what vitrification media they're using is trehalose and glycine, and also they add a human serum albumin too. So when they made vitrification media, they add to the semen sample slowly, just like I mentioned before, just like doing slow freezing. You add it very slowly, stepwise, and mix it thoroughly, and incubate at 25 degrees for 5 minutes. Then you start load to the vials. You can see there's a kind of funnel shape. So you put you put the whole thing to the liquid nitrogen. Liquid nitrogen they use as a medical grade, like liquid nitrogen is free from the contaminants. When they load about 25 microliter of the semen and drop it directly into the cryo valve. And then they keep doing that until they collect enough sample of the semen, and then they transfer to the liquid tank. All right, next slide, please. All right, this is a sperm VD. That's our favorite device in it's very common, or I think a majority of sperm vitrification in the United States are using sperm VD. It's very efficient medical device for cryopreservation small number of sperm. Next slide, please. All right, so this is the first step, and you can see there's a dish we prepare for the sperm search plate. You can see on the left side there are microliter droplets. That's for we using for sperm searching. And in the middle top, you can see the white droplet. That's a PVP. The PVP using for when we collect the sperm, if it's moving, swimming really fast, we put in the PVP, slow down the sperm swimming, and it's easier for us to use micro manipulator to catch the sperm. And in the middle, you can see small 3 drop. So the reason why we have 3 drops, the first one we use to collect the progressively motile sperm. In the middle one used for collect like locally moving or twitching sperm, and the bottom one we collect for like immotile sperm, just like a TASI. A surgery sperm, it's usually most of our sperm are immotile, so we collect for the bottom drop. And also you can see the right side is blank. That's for in the future later when you have a sperm VD, so we put there. Next slide. All right, so this is how you search for spermatozoa. So first you load your sample to the drop and you put in the microscope. You usually start from the border, the edge of the drop, because if the sperm swimming, they like to go to the swimming to the edge of the sperm for fresh media, or or fresh oxygen or something. So they like to swimming to the border. You search for them, you collect them, and then you move those motile sperm to the top drop. And then next step, you start searching for those locally moving sperm, like twitching, and so that won't swim to the edge. So you search from in the middle, and also this sperm you collect it and then you put to the middle drop. And then if it's most of a TASI sample, most are immotile, but they are sperm, so you collect and put in the bottom. Also, they recommend use a larger diameter of micro manipulator to collect those to reduce the risk to for the damaged sperm membrane. Also, the larger diameter helps to prevent clotting as well. So then next slide, please. All right, then we're going to transfer spermatozoa to sperm VD. Sperm VD, you load a sperm freezing media and wash media. It's about like 1 to 5 microliter. You can see the right side, that's a sperm VD device. Once you collect all the sperm in the middle 3 drops, then you use a micro manipulator, you catch them and you transfer to the small droplet in the sperm VD device, small 3 drops there. Once you have it, so you have to freeze them within 8 minutes. Begin the excess not used for egg seed, because you want to save as many sperm for the future egg-seed case. All right, next one. Next slides, please. And then, oh, sorry, this one, yeah. So you gently pick the sperm VD from the dish and transfer to a cryo vial. The cryo vial, you label your patient information and the date of the freezing, and then you put in you plunge into the liquid nitrogen tank. You don't have to put the vapor phase because this is vitrification. You go straight to the liquid nitrogen. This is the most delicate phase because when you place the cryo vial and the rack or lowering the rack into the liquid nitrogen, the droplets could move around or something, could be dislodged. So you have to be really quick, but have to be very stable. Try to avoid, all of a sudden you shake a little bit or something, you may lose the sample. Also, when you leave excess oil to kind of protect it's kind of another protection layer for the droplets. The other tip is when you freeze into the cryo vial, you don't tighten the cryo vial to leave, is to lose the cap and let the vapor of liquid nitrogen go inside, just in case in the future when you take out and so still has a liquid, the vapor of the liquid nitrogen can protect the sperm. All right, next one. So this is after vitrification. They recommend the warming method. So remove the cryo vial from a liquid nitrogen, unscrew the cap, and let it thaw in horizontal position at the room temperature or a heated surface. Usually it's like 37 degree or 42 degree until the oil cover the droplets liquefies completely. And then you remove the sperm VD from the cryo vial, place into the egg seed dish. You can see the right is egg seed layout. So you use a micro manipulator to catch the sperm from the sperm VD. And then you move to the middle one. There's a sperm wash media. You rinse it over there. And then you collect them in the middle drop for the future egg seed. All right, next slide, please. So that's the warming protocol I just mentioned is just for sperm VD. There are a variety of sperm warming protocol. So some lab they're warming at the room temperature, some warm at the 42 degree, or some they warm at the 44 degree. So I've been seeing research talk about when you warm at the 42 or 44 degree, actually have a better recovery rate and a better motility recovery rate compared to the room temperature. So they recommend if you do warming temp. So some lab they recommend warm at the 42 degree, have a better recovery rate. Next slide, please. All right, so once we have a vitrified sperm, so we need storage, right? So usually we put the liquid nitrogen tank that's minus 196 degree. Also, vitrified sperm can be stored in the temperature minus 80 freezer. That simplify the storage and save a lot of the space too. Also, there's a research paper talking about the lyophilized sperm Zola. That's another method, but once the sperm has been lyophilized, they lose all the motility, so they don't move at all. So once you thaw, the only usage is you have to use this egg seed. You can't do other method to do the egg seed or something. That's the only thing you can do. All right. Next slide, please. All right, so when we talk about the cryo storage, we know that liquid nitrogen, especially unsterilized commercial liquid nitrogen, and has a large variety of bacteria, virus, or fungi, and can cause the transmission or propagation of the disease. There's no absolute way to say that contamination risk is free. There's nothing, there's no method exists. So we just need to minimize the risk. So they have been found in the bottom of the storage container. They're all different kinds of bacteria. Also, bacteria has a higher tolerance to liquid nitrogen compared to the virus and the fungi. Usually, fungi is not that common, but bacteria is very common. So the common method to reduce the risk, most of it using the filter with 0.22 micrometer to filter either the liquid vapor or liquid nitrogen to create a small, either the liquid nitrogen vapor or liquid nitrogen droplet. All right, next one. Next slides, please. All right, so that's about today. I talk about the sperm slow freezing vitrification. So now we know sperm vitrification provides a simpler, faster, most cost-effective alternative to conventional method. But you still usually it's for patients have a small number of sperm. So in the future, still, this hasn't been validated method. So future direction for the research, we're thinking about could be validation of this method, whether, this sperm can improve the clinical outcome. Because for our clinic, we don't have enough cases to do research now because, in the past 3 years, the total case are about 20. But I think if it's, whole country, probably more. I think probably next 4 or 3 years, so maybe we have more cases, then we can start conduct the study to see if the vitrification of the sperm can improve, the fertility rate or embryo like blastocyst rate or pregnancy rate or live birth rate compared to the slow freezing method. All right, that's it. And then thank you everybody. Sorry for the beginning. We have all different technical issues. Sorry about that. All right, any questions?
Thank you, Dr. Feng Gao. It's a nice webinar. Any questions? This is open for these discussions. Any questions from audience sides?
Yes, sir. How long do sperm live?
How longer?
Yeah, speak loudly.
How long do sperm live?
How longer stowing? How long do sperm live? Oh, you mean like outside once they collect the sperm?
Yes, ma'am.
Oh, so you mean usually once the patient collect, usually if they don't have a problem with collection, once they collect the sperm, usually we keep in the 37 degree. Usually takes us about half an hour liquefaction, and then afterwards we either wash the sperm for fresh eggsy case, or if we do sperm cryo, we do right away. So you don't keep the sperm long. usually within 1 hour, you have to do something. Does that answer your question?
Yes, ma'am.
Oh, thank you.
Thank you.
Mhm.
Anyone?
Also, I want to ask, are there any embryologists in here? How
Some of the
often do you
questions
guys use
box.
sperm vitrification?
Seeing some of the question in a message box. Can you read it?
Okay, what's the main reason for infertility in male? Oh, okay. What's the main reason for infertility in male? So I don't know if you are keep track of the sperm in the whole world. We know that the infertility in male, the problem is keeping growing and the sperm quality going down. That's happened all over the world. What's the main reason? So for us, if the patient coming to our clinic, usually most of the reason for infertility in male is oligospermia, like a low concentration, low sperm motility. I think the main reason is either the pollution or like smoking or alcohol or something. I think that's the reason for infertility in male. Well, also depends on also they have a genetic reason too. Next question is how many sperm will reach the egg? Do you mean like a conventional insemination or in natural in human body? In human, but naturally in sperm, you have like 4 million sperm or something in once you ejaculate it, but only several hundreds sperm will reach the fallopian tube. That's where the egg meets the sperm. That's the natural. So for the conventional IVF, what we do is we incubate egg with the sperm in one dish. The concentration of the sperm usually is like a 200K per mil. We incubate them together, and then hopefully they can have probably 1 or 2 go inside of a egg. But once the sperm go inside the egg, there's a blocking system. Only one sperm can go inside. Once the sperm goes to start penetrating sperm, the egg start activate the blocking system to prevent other sperm going inside. All right, so link is valid. Is that link for the sperm VD? Are you guys talking about? Oh, okay. Yeah, the sperm VD in the YouTube. Sorry, go ahead, please.
Yeah, one question. As you told that lyophilization of spermatozoa is another method. So is it possible for retrieve the vital sperms and oocyte fertilization?
The lyophilized sperm is very rare. Not many paper available, but it is I think at least I saw 1 or 2 paper talk about the lyophilized sperm, and I saw the paper they talking about once you warm the sperm, they lose all the motility. So only you can use for egg seed.
Okay.
I think a lyophilized sperm is not the ideal way to store the sperm, but I think if you don't have other options, or it's easier for transportation purpose, but I think it's not common at all.
But
we never did that.
If the patient is with a severe oligospermia, what we will do for that?
What do we do for that? Egg seed. Usually in clinic, so we recommend do fresh sperm and collection, and we do egg seed. But if sometimes the patient cannot, for example, their business trip or something, they can't make it on a day for his partner or wife for the egg retrieval, so we freeze them. That's why we do the sperm vitrification to store their sperm.
But have you recommended for the DNA fragmentations to this severe oligospermia patient?
Yeah, we do. Yeah, because there are also different reasons for severe oligo. Is there genetic reason or different DNA fragmentation rate really high or something? So it depends on. So we usually recommend patients do that fragmentation test.
Okay.
So in our clinic, usually we strongly recommend patients come to collect the fresh sperm. Even you have oligosperm, we still can pick the from the small fresh sample to get the enough for egg seed. But if you cannot make it, so then probably sperm vitrification is a choice.
But after the thawing of the samples, sperms, what is the success? What is the fertilization rate?
So for the sperm VD, it's about 90% retrieval recovery rate. That means we can find most of the sperm, but it doesn't mean all the sperm can be revived. Motility or something lost. So I think only like 1/3 afterwards we can use it for egg seed or something. In general, like slow freezing or all right, so sorry, go ahead. Oh, I thought he has a question. Sorry.
So what is the pregnancy outcome?
For sperm vitrification?
Yes.
I haven't seen many study about this because it's not very common method. So in our clinic, in total, we only have maybe 20 to 30 cases for sperm vitrification, and some we haven't even saw them yet. So this is for storage purpose or for future case. I think that in next few years, once we have a more sample, and once they have start doing egg seed or stuff, and once we have a data, we can start compare the data. But I think that if you look at the literature, probably there some research showing that vitrification sperm actually better recovery rate and a better what's called mitochondrial activity recover, and also different the DNA fragmentation rate is lower compared to slow freezing. I don't think there are a lot of studies about the pregnancy, but they do compare fragmentation rate and the mitochondrial activity and stuff like that. Also, I think some research doing epigenetics too. They want to know after vitrification is going to change or not. Still not very, not many study about this yet.
As you told that the possibility that vitrified sperm will be stored in a -8 degree. -80. So how many days we will store it?
That's I just put in there because that's the review I put in there. But I actually, to be honest, we don't store in -80. We put in the minus. You put a liquid nitrogen tank. That's minus 190 degrees, 96 degrees. That's you can store for forever. Minus 80 degrees, I doubt it, because not many validation study yet. So we don't store in the minus 80 degree in our lab.
Okay.
Yeah, to be safe, I still strongly recommend to store in the liquid nitrogen instead of a minus 80. All right, so let me read the question in here. Is vitrification better than slow freezing sperm? Is it preferable? So that's why it depends, because the slow freezing sperm is easy to do, and then it's a routine schedule, so you don't have that micro manipulator be involved. Slow freezing, you just add a media and then you wait and that's it. But vitrification, you need a lot of people be involved, like making dishes. You have to someone to micro manipulate or catch it. But if the patient has oligosperm or azoosperm, they need the storage for now because they can they want to store the sometimes like a cancer patients, they want to preserve their fertility now before their chemotherapy or something. So they have to preserve all the small number sperm, so they have to use this vitrification. For the normal patient, the regular patient, if they have a normal sperm, so you don't have to use the vitrification. Also, vitrification looks like they have a better recovery rate, and also the motility, like I mentioned before, mitochondrial activity, and the DNA fragmentation rate is lower than the slow freezing. Method itself is better, but it's a very complicated procedure. Yeah, that's, I think also I talk about advantage, disadvantage. Vitrification is that it's efficient, but it's a lot of steps. How can sperm stay frozen? Once you put in the vitrified liquid nitrogen, it can stay there for, I think,
That
for
depends
forever.
on the level of the liquid nitrogen. So
Also, well, you don't want your liquid in the nitrogen tank is low. You probably you have heard of a lot of incident in United States. Yeah, there's an automatic refiller for liquid nitrogen tank. Actually, it happened not our clinic, a different clinic. It's not far from our clinic. It's in San Francisco. It's a huge suitcase because liquid nitrogen level is low, and then embryo and sperm or something quality compromised. A lot of patients sue that clinic. So you have to make sure liquid nitrogen tank. We check every day. We measure the level every day, and then every other day we refill it. All right, any more questions?
any questions from audience? Or else we will end of the section. Any questions?
All right, thank you so much.
Thank you. Thank you.
Yeah.
Thank you. We will end of the session. So thank you, audience. Thank you. So as we come to this conclusion part, finally we will be enlightened and enriching the webinar. And first and foremost, I will express our deepest gratitude to the estimated guest speaker, Dr. Feng Gao. Dr. Zhao is expertise and insight to our truly invaluable, and today's webinar is really grand success. And your passion in your field is dedicated and advancing and scientific knowledge. So that is very impressive. And this is my field also. I really dedicated to saw this, and this knowledge have really remark with us, and we are grateful to us knowledge and inspirations you have shared with us today, and all these participants, 90 to 93 participants consistently waiting for this today's webinar. I would like to extend all these participants also join today and see means learn to this vitrification procedure. There is a very innovative subject. If you are in a fertility field, so that is very crucial, and the pregnancy outcome. And I also thankful to the all these participants and the organizer and everyone to this success, grand success. Thank you, Feng. It's nice, and for the future we will try to collaborate any things. We will write to the papers, and we will try some of the experimental things also. So once again, thank you for being a part of this webinar, and your presence has made a significant difference, and we hope to see you again in the future events, and any of these scientific things related to this genomics as well as for this fertility thing. So we will be things. Wish you all this wonderful day ahead. Thank you.
All right. I really enjoyed talking to you guys, all of you. Hope you have a great day.
Thank you. Thank you. For,
Yeah.
you are really, you have a given for the valuable times today. And this is right now, what is the times? 10:30,
12
you know?
o'clock. 12
12.
o'clock, 12:06 in the morning. It's a Friday now here.
Yeah,
I started
yeah.
Thursday, now it's a Friday.
It's an Indian time, so it's around 12:30. And your side, you have night note to 14-hour difference.
Oh, all right. Yeah. All right, then. Probably we'll see you next time then. Hopefully.
Thank you.
Yeah.
We will be try to connect you, and when I will going to the San Francisco, I will definitely call you, and we will
Yeah,
meet.
please visit. Yeah, visit us. So yeah, of course. Yeah, welcome everyone. All right. Yeah,
And
have a great
audience,
evening.
everyone's to Friends is inviting us, and when you will be going to that sites, and you will meet
Yes.
once you heard, and I will share to any of the questions in your site. I will share to Dr. Feng's to mail ID, so you will ask directly to her also. Okay, Dr. Feng's, and I will share to your mail ID to everyone.
Say to
Anyone
your.
to ask any questions, so they will be directly mailed to you.
There will be. Who? Oh. Thank you, ma'am, for. All right. So that's it, right?
Yeah. Thank you. Thank you, Pin.
All right.
So, yeah, thank you. And on behalf of the audience, I really thankful to you to join this event, and wonderful webinar, really amazing webinar, and it's a successful webinar also. So thank you very much. And
All right. Thank
now
you.
you will.
Bye,
Thank you.
everyone.